This week is about engineering immunity. I think it's kind of one of the most interesting and important field, I guess. So the topic is engineering immunity. So here we want to understand first the basics, immune system, and especially a little less covered topics, so-called lymphatics today, and role of vaccines and the antibodies in disease prevention. That's what we will cover today. So immune system and lymphatics, I'm assuming you all have watched the video, so we'll go a little fast and let's try to focus more on your questions. Okay, so we all know that cells of the immune system, the key part is distinguishing self from non-self. And how do we develop this a vast number of antibody diversity? This is a separate topic in just you have to take immunology course. But our body, when we develop the immune system, it's just a fascinating ways how to discriminate a self from non-self. Non-self even includes not our body, but even like your friends or other human bodies as well. Okay, and what's interesting here is for the one of important route for our immune system is the lymphatics. And lymphatics also have a capillary. So you can see our typical circulation, we see vessels, but you see also there are actively intertwined lymphatic vessels. And interesting, that's kind of one open-ended, not like a circulation, a closed loop. So that's a big difference. And you can see eventually, whatever inject your body, like let's say, if you inject a dye molecule into your muscle, they will be drained through these lymphatic capillaries. And in the end, it goes combined to our venous system. In fact, honestly, I haven't really seen this combination point. So if there's anyone who actually watched this, let me know. I'm curious about this. So first, we call blood plasma and serum. I think people have seen like how to transfuse. There's war ongoing, and if there's war and injury, if a person loses blood, and that's gonna be a big problem because you know our circulation, and you don't want to lose too much blood volume so that you have a lower blood pressure. It can give the patient into a coma, so which is not great. So these yellowish ones are actually blood plasma, and liquid component blood, white blood cells are suspended and for transfusion. So that is called Yundami.
Plasma.
Exactly. If it's a plasma, you want to take out clotting factors such as fibrinogen. What do you call it?
It is serum.
Okay, that's great. That's called serum. Maybe serum is more known in cosmetics industry, but for those who have done any cell culture, you want to add a serum such as bovine serum, albumin, and those serums you want to take out the clotting factor. So when you like centrifuge blood, that's how actually we make a plasma. So you can see, depending on the density, you will see RBCs. And also there's a way to select, take off this white blood cell, which is called a whitish buffy coat. And that's a very little amount, but this is where the leukocytes, so-called white blood cells and platelets are there. And the remaining is called a blood plasma, and which composes a lot of factors, proteins, and even antibodies are there too. All right, so there's a question by Uju. There's Uju. When we do experiment with blood for immune analysis, such as cytokine analysis, which type of blood is preferred and why? Uju, can you explain which type of blood is preferred? What do you mean by this?
I mean, when we do experiment with blood, we don't use usually the whole blood, so we just separate plasma or serum. So I mean, which type of blood? I mean, the plasma or serum.
Okay, yes. So you're asking whether it's whole blood or plasma we're gonna use. So I guess that's probably related to whether cytokine assay is for cytokine. Cytokines are inside blood plasma rather than probably not inside RBC. So that probably is what you are asking. And not, I can't see your face. I guess you have some reason today. How can you extract plasma for given blood plasma bag? And I think that's something I already showed you, like centrifugation. Based on that, we can actually separate them. So for this, I guess, I think I want to share some of this, because I think these days cytokines is very, very important. Everyone hear about cytokine storm. So let's go through a little bit of a review for that. So first, as Uju asked about cytokine, it's a general term for small secreted proteins, and this is a key modulator of inflammation. And so before we getting into cytokine assay, which Uju asked, but I also know in even our labs, students are working on cytokine assay to figure out the inflammatory response of a body with respect to other outside stimulation or modulation or treatment. Okay, so you can see these, what does the cytokine do in the body? So cytokines, cyto means a cell, right? So it's affect growth of blood cells and other cells to help eventual body's immune and inflammation responses. Okay, so cytokine, what kind of cytokines are there? And cytokine is a large family of molecules in this case. And of course, we heard about cytokine related to COVID, right? And why COVID infection has been very, very dangerous is because of the aggressive inflammatory response that is due to the release of large amount of a pro-inflammatory cytokines that we call cytokine storm. So that host response has been critical for devastating initial outcome. Right, so for that, and for that reason or another reason, there are probably many assays for that. So you can see in Google first, like assays advertisements are coming first, but let's see. So what the cytokine assays Uju is describing is an assessment of preparation, treatment of blood and tissue samples related to cytokine. So Uju's question is more about the blood sample, but actually even in our lab, we also take out the tissue and then grind it and then use that supernatant of tissue, we use it for cytokine assay. In addition, so we don't know whether this tissue is a more localized response of our interest, but on the other hand, we are also interested in the systemic, the whole body inflammatory response. So we can also draw blood and we can use that blood and especially blood plasma for cytokine assay. So that's what I hope answered Uju's question. So why this is important? Essentially in disease diagnosing and monitoring, because cytokine can be an important player in the progression and regression of pathological process. So in a nutshell, cytokine can be a very nice biomarker for many diseases. So what are the biomarkers? We call biomarker all the time, and this is very important concept. So biomarkers is whatever aspect of a living body and which represents the, let's say, the disease status or even treatment status. So monitoring biomarker is a very important concept. So cytokine can become a kind of a biomarker. For example, even like imaging can be a biomarker. So imaging biomarker. Or in pharmaceutical companies, what are the most important biomarker is actually monitoring biomarker, which actually show for, let's say, if there's a Alzheimer's disease, we are trying to develop a treatment. And how do we know that the treatment is being well done, well going or not? We do need a disease monitoring biomarker. Okay, so in that context, a cytokine can be a very important biomarker. So then we have to measure somehow this. How do we measure? A good way is immunoassays, because it's basically immunology aspect. So immunoassays, especially so-called using ELISA. Those who are familiar with this are there, but there are also people who may not be familiar. We call simply ELISA. It's enzyme-linked immunoabsorbance assay, and that is using primary antibody for capturing the antigen, and you have to amplify it for proper detection. So you need a secondary antibody conjugate to an enzyme to make a pigmentation or radioisotope for radioactive detection. Okay. So in fact, when you are doing research, you actually want to figure out what cytokines are actually involved in a disease process or a treatment process. Then how can you find that out? But we have to know there are a bunch of cytokines. So just simply, there are pro-inflammatory cytokines and anti-inflammatory cytokines. There are a number of them. So in fact, you rather use simply one aspect, you want to use a panel. So that's why cytokine panel assay. So this panel you want to use to identify the levels, especially elevation of specific cytokines, whether it's pro-inflammatory or whether it's a anti-inflammatory. So in that regard, so when you are doing research, I think this is very useful, not only for COVID research. And this, I think, ELISA, I already explained, so I think this will be okay for now. Let's say, how can you extract plasma for a given plasma bank? I think I prepared one here for how to prepare blood plasma. Usually this is a blood transfusion process. So let's say you are in a workplace and you have your colleague or your comrade just fell down due to like bleeding. And what can you do? So for that, you need to have a plasma prepared for transfusion. And a good thing about this is you have better to freshly frozen, called the FFP, fresh frozen plasma. But you have to separate RBCs and platelets from whole blood. How? By centrifugation or by apheresis technique. And you have that, and with this, you can only administer intravenously. So this is quite not easy for, I guess, typical people, but also important things, we have to make ABO compatible. So if it doesn't work, you've got a big issue, right? So you have to know the recipient's RBC types, blood types, and then you can actually use it. So hopefully this makes good sense. And let's move on to, so what happens to cellular response to inflammation? So I happen to actually have this experiment myself. So I labeled RBCs with a fluorescence. So, and I used developed a video ray to photon microscope to observe how RBCs are moving, and by making labeling RBCs, but happened to be there's white blood cells are also labeled. So let's take a look. I think this is a schematic how it works. The quality is not great, but this is what you see these big cells on the wall. These are white blood cells. And these tiny, fast moving ones are RBCs. Let me just replay so that you can actually take a look at it again. So you see a fast moving, and those big cells got stuck in this blood vessel on the wall. These are white blood cells. And that's all, of course, very important for inflammation. So, and the other thing is, like, unlike blood vessels, lymphatic vessels are another kind of very important in our immune system. So for observing lymphatic vessels, we have to inject contrast agent into the tissue. They will drain and then get filling the lymphatic vessels here. So what you are seeing is negative contrast for the cells. So this is lymphatic flow. Lymph nodes do not have a heart or pump, so it has to depend on passive way. So in nearby, like pulsation from the arteries, lymphatic. So that's why it has this a a very, you see a cell clump here, and I want to just show that this way. Let's see, you wait, this cell clump may leave out here. Finding this out is very difficult, but this is kind of a demonstration and showing how in vivo imaging may reveal how a lymphatic metastasis can happen. Okay, so that's an example. So you see, I did this work. And cellular response to inflammation. So I'm going to just show you with one movie. It's full of terminologies, but I think this is important to understand a little more detail. So let's go to this. Let's move on. Sorry. It doesn't work.
I am the Terminator. I have been sent back in time to defend John Connor from infection.
So
I have occupied this Phil for transport.
The first
trapped in the blood vessel. Inflammation.
From neutrophil.
My ally is the mast cell.
Inflammation.
Secreting his histamine.
The
blood vessel.
And they will dilate the blood vessel. So called vasodilation. It's one aspect.
Blood flow is slowing down.
Blood flow
this will
slow
aid
down.
My escape. The endothelium is releasing
the endothelium nearby will now express.
Now I can bind to the wall using my cellular Lewis X module.
Slow down and
but the attachment is weak.
And attach.
I will roll, but I cannot adhere. I have failed.
But it's not enough
the
to adhere and come out.
Will help me.
But inside
macrophage
cells,
cytokines. IL-1 alpha CXCL8. CXCL8 it binds to the endothelium. I can use it to bother my cause.
And then
the other cytokines, they have stimulated production of E-selectin. I can roll better.
They will grab this white blood cell.
Yes, at last they have produced ICAM as well.
And I
but
can
I am not yet ready to use it.
Oh yeah, no.
CXCL8 is the key. I can use it to signal
they were
integrate
and
command it to grow in power,
in
affinity.
The white blood cell will now have a high affinity. Will change. It changes white blood cell phenotype.
Now to find the landing site.
Will bind.
These will do.
Correct.
Attachment.
So that's a disease molecule.
Plate. On to the final phase of the blood.
Then what happens
is
there are multiple
backup guide us through the wall.
To make the cell come outside of the vessel. So diabetes is important terminology.
We will be
So
free,
that the white blood cell
free to exact vengeance on the infection.
Okay. Okay, let's see. So the reason why I watch the video with you is because the next. So all the details, in fact, is a very important immune cells. So when inside the lumen, vessel lumen, they roar, and from this signaling from local inflammation, they will start to roar and tight binding based on involved with multiple molecules, and they crawl outside called diapedesis, or you can call this as an intravegetation of the white blood cells. and they start to migrate towards. So involved molecules are here, ICAM1 intercellular adhesion molecule. And also you saw that there's integrin molecule, which is very important to originally attach it to extracellular matrix, such as fibronectin for the cell. So this is what you watch is neutrophil extravasation from blood vessels to the site of tissue injury or infection. And this we call innate immune response because this is non-specific, but it's very important at the first line of defense system for our body. And here, one thing you should remember is the term diapedesis, which means leukocyte extravasation or adhesion cascade from the inside blood vessel to the outside tissue. All right. So this immunological system is the key organ is a lymph node actually here. And you can see we have a bunch of lymph nodes. So you can even notice if you have an infection, you can kind of touch and find out enlargement of lymph node. Okay. And of course, inside lymph node, there are many immune cells are residing, waiting for the external pathogens are coming in. They want to detect it here and to do proper immune and inflammatory response. So B cell and T cells are actually residing over here. And there are blood vessels, of course, and blood vessels getting in is called afferent vessels. Getting outside of this is called efferent blood vessels. So here comes the Anho's question. Okay, so what's the difference in detecting the flow in lymphatic vessels and blood vessels? Okay, and how can you apply for lymphatic image? So a very interesting question and important question because we all are mostly interested in seeing blood vessels so far. But in fact, blood vessels we've learned from cardiovascular system. It's just part of 1 out of 4 a big physiology topic. But lymphatics are not yet because I think we don't know that much. So it's a less explored area. How at the same time, lymphatic flow is not like that dynamic like cardiovascular blood flow because it is passive. There's no pump. However, so it's passive. I think personally, I see those who are doing stretching or yoga or any kind of exercise, actually you're boosting your immune system by making the flow of lymphatic better than just sitting, staying, not moving. Okay, so and there is another very interesting question by Janice about thymus gland stop to compromise immune system. What will happen to human body? So where's Janice? Oh yeah, there it is. Yeah. Yeah, it's a quite advanced question that Janice knew that thymus gland is important immune system. I just talk T cells, and that T comes from thymus, in fact. Okay, so in the laboratory mouse, especially for those who are working with a human cancer, implanting into a mouse to grow, to test new ways of cancer drug development. But that uses a nude mouse. It's also called a thymic nude mouse, meaning that this mouse somehow transgenic and it doesn't have a thymus, meaning that no thymus means no T cell. So T cell is lacking in this mouse, so it's immune deficient mouse. So likewise, human may have this issue, which I think I searched it a little bit. So important thing is our T cell development and B cell development is actually from the bone as a developmental process to make all our antibody and also T cell receptor diversity at the early stage. So after we grown up, I guess even if we damage our thymus, maybe our immune system may be okay. That's my guess, but I'm not a expert immunologist, so do not believe it totally to me. Okay, so that's my guess. So let's take a look related to this. The first, lymphatic. So lymphatic blood vessel imaging. So this is a very hot research area because we haven't explored it much. So what are the lymphatic imaging? Is the conventional imaging like CT or MRI. You can actually do it for enlargement of lymph nodes because that's a diagnostic criterion for certain type of disease, especially like affecting our immune system. And can MRI see lymphatics system? Yeah, to assess lymph node because lymph node is rather big and central and peripheral lymphatics. So MRI is a quite, I think, a good aspect. But there are also CT and ultrasound can be also used to evaluate lymph node. So here, real lymphatic vessel is small, and also lymphatic capillary is like blood vessel capillary, which is not even easy to see. Not only with microscopy, yes, but clinical purpose, it may be very hard. But there are specific terms for that. Lymphangiogram, meaning that lymphatic blood vessel see. How do you do that? So it's interesting for people who are suffering from, let's say, lymphedema or problem with the circulation. We may want to check that the lymphatics are working well. So how do we do that? It's actually easy. You just inject a dye molecule into the, let's say this is food. You just inject anywhere in the tissue. Why? You don't need to get into blood vessel. It drains through this lymphatic system, and you can actually, if it has a contrast, you can actually see with an X-ray. So healthcare provider, your feet inject dye into between your toes, just inject it into tissue, it'll drain, and that drained lines are actually showing up your lymphatic vessels. And like you want to see the timing and how they flow actually shows the wellness of lymphatic flows, so that identifying lymph channels, also identifying lymphatic vessel, lymphatic circulation or lymphatic flow, which will be very important in clinical aspects. All right, so yeah, I just searched a little bit of the imaging techniques to show how lymphatic vessels can be imaged. This is one nice image showing that blood vessel, you have to have a contrast agent in blood, but lymphatic, you just inject contrast agent into the tissue. Just for drainage, you can see this lymphatic vessels as well. It's very interesting. And also this cutaneous, so in your skin, there's not only this red blood vessels, there are this green annotated lymphatic vessels as well. So you can see this 3D microstructure, which is fascinating. So we have plenty of lymphatic vessels all together with the blood vessel. And hopefully this one gives you an idea how important it is. Okay, and then the thymus aspect. So I think that's a, yeah, Janice asked a very fascinating question because thymus is this organ. And when damage is done to injury repair in thymus, okay, especially, I guess, yeah, more practical issues when you're a cancer patient, ionizing radiation on the chest can damage the thymus. So what would happen if your thymus is damaged? So removal of an organ has a little effect. Dirt has a little effect. So you see, thymus might not be that problematic for adult, but when the newborn, when your immune system is not well trained yet, the damage to thymus will become a big issue because the T cells in the blood and lymphoid tissue, such as lymph node, are depleted, and the failure of immune system causing a gradual and fatal problem. So of course, thymus is very, very important primary lymphoid organ. And why this is important, and we are not covering this class, but B cells, which are supposed to produce antibodies, and T cells are active in adaptive immune system, which T cell receptors and B cell antibodies, they are at the beginning of a life development. they are like generating a vast amount of diversity, which if they recognize self, they will be dying by undergoing a programmed cell death or apoptotic pathway. So only the cells will recognize outside of our body will remain. That process we call maturation. And especially T cells will mature inside the thymus. Remember the term called a niche concept, stem cell niche. So that stem cell develops in the thymus, and that specific three-dimensional structural and biochemical cues, they will mature into a matured T cell, but immature lymphocytes are differentiated into antigen-sensitive lymphocytes. And so if thymus are removed from your body, your system becomes weak and prone to infectious diseases, which will be a big problem, but not after you become an adult, right? So I hope this one answers Janice and everyone's question. So let's get into the engineering immunity and vaccine. There are so many different kind of vaccines, and this is already, I think, like old ones. So now I think we have a better idea and better vaccines, especially in the past couple of years, we've observed an amazing, a new innovation in terms of vaccine industry that now we are developing mRNA vaccine, which is, I guess it's a revolution in human history. But the goal of engineering immunity and is to enhance our function of immune system, which remain true. And that's why we develop vaccine. And the vaccine is agent to stimulate body's immune response by recognizing the agent as, let's say, Kim Young-min. Young-min?
Yes, Professor.
Yeah. So
Sorry.
exactly, that's a vaccine, right? And many vaccines are used to stimulate production of
Antibodies.
great, yeah, and that provide immunity against a particular disease. We're talking particular disease, so that means we need to have a different kind of vaccine because our immune system, especially antibody, is a very, very specific. So regarding to that, there are interesting questions by Janice. In case of injected vaccine, but there's not stimulate immune system of body, is there other vaccine for use instead of inactive vaccine? So it's a little bit hard to understand, but I guess what you mean is even you inject vaccine, sometimes it doesn't work. That's true? Yeah, and of course, there are different kind of vaccines instead of inactivated vaccine. And I guess that kind of go along with Jung-Yoon's question. What are the differences between antigen-based vaccine development and mRNA vaccine? I think it's a pertinent question because this is something you can't really find it textbook right now. So Mahunur, I think, has a very good answer. So I'll go through with her. Antigen vaccines, like conventional ones, contain pathogenic component, which either called the viral or bacterial, that can be the surface proteins or the genome, whereas RNA vaccine, the new type of vaccines, are specifically contains only mRNA. You guys remember mRNA, from there, after translation, it becomes a protein, right? The former, the antigen vaccine is a slow. Remember how difficult is it? I think it's a next Wednesday class, how to produce a conventional vaccine, because you have to produce cell line and extract and gather all the antibodies. It's just very time and cost is like enormous compared to the mRNA vaccine or made from DNA template directly. So that's a difference. Antigen vaccines directly stimulate. So you give out the antigen directly into the body for the immune system to train, while RNA vaccines, RNA is not protein yet. So it enters the inside our body host, and it has to enter into the host cells to manufacture these antigen or epitopes and give instruction for production of antigen to stimulate the immune system. In fact, what I remember is the problem of delivery of mRNA into the body, let's say into the circulation, it doesn't work very well. So innovation comes with the drug delivery system like liposome, how to design those so that it can safely deliver mRNA into the host cells, so that has been innovation in the recent years, so that's how we actually can produce a lot of mRNA vaccines, which I also got it like 2-3 times. Okay, so let's move on. This antibody structure, which I think many of you know already. So let's see. Shin So-yeon. So I said like antibody is a Y shape, but this is just a kind of a topologic way. They don't look like Y, okay? They look like more like ball. So that's why we call immunoglobulin. Globe means a ball, okay? However, like this antibody recognition, that substance can be recognized by antibody or immune cell receptor is called?
Antigen.
Exactly, antigen. But this is an antigen, but this part of antigen recognized by specific antibody is also called antigenic determinant, is called? Exactly, epitope. Epi means a surface. So you can see even one antigen, there can be different types of epitopes. Okay, so that's where we distinguish. When we produce antibody, we could produce, like for this antigen, we could, the body will produce multiple type of antibody we call polyclonal antibody. While for therapeutic purpose, like very specific, only like this purpose, only single type, we call a monoclonal antibody. So as I said, antibody, there are other words for that. Let's see. 정우주?
It's the immunoglobulin.
Exactly. You know, globe is globe, right? Globe. Okay, globular plasma protein. And that antibody is produced by Uju?
It's plasma.
Exactly. Yes. Plasma cells are coming matured from B cell. Okay. And that B cell is matured. So this immune B cells from where?
From bone marrow.
Exactly, bone marrow. So inside the bone, right? So that's why bone marrow is very important. And as I said, specific kind of antibody by a single clone of cells or cell line, and they always bind to exactly the same epitope and exactly identical. That's called?
It's monoclonal.
Exactly, monoclonal. So when you actually look at the drug name, you watch the drug name, the last one, it ends with the MAB called the MAP. When you find MAP, you directly recognize, hey, that's monoclonal antibody drug. Of course, antibodies are very, very expensive. All right. And the structure of antibody, you guys are all know in the basic long chain and short light chain. So long chain called heavy chain, light chain, and even a single IgG molecule has 2 antigen binding site. And this production of this is just an amazing engineering process made by nature, I guess, because it's a hyper variable region called the CDJ joint makes of like virtually whatever in the universe you can actually detect. So that is the beauty of this. And we call that as a CDR, or let's see, 정윤.
Complementary.
Exactly, complementary. Why? Because it's complementary to the antigen itself, okay? And the important thing here is FAB, or fragments of antigen binding site, is this part, and the other root part, it's constant, so that's called FC region. And remember, the antibody typically is a pretty big molecule. So typically 150 kilodalton, and the size will be about 10 to 15 nanometer. All right, so here regarding that Uju's question, detecting COVID-19, we often measure IgG ELISA kit. These days, yes. Is there a significant correlation between IgG and coronavirus? I think it's a very interesting question. And I think Mahnur had a very good answer. So I think we can go through here. So IgG, which we will cover next slide, but immunoglobulin type G is meaning that immune system is functioning properly. So IgG is the most abundant antibodies in our body. When there is an infection, so IgG is the first line of defense. But it doesn't say it has a specific relation of general observation in infections. One thing important is, like, we know that this IgG-based ELISA kit is a little less specific, and even if it gives a negative, that doesn't mean that you don't get infection. So that's why we move on to PCR test for further very, very sensitive testing. But it's the important thing is, if we have a less IgG in our body, there's a high chance for us to get infected. So that's a very important aspect. All right, so here comes the classes of antibody or immunoglobulins. There are so many, like 5 times. Even I don't remember everything. So I just want to point out what matters to us to understand and remember. So IgA, D, E, G, M. And what matters is in biochemical aspects of this very interesting protein molecule is strength of a single interaction of protein ligand binding that we call, let's see, Anho?
Affinity.
Exactly, that's called affinity. But you can see here, there are 2 binding sites. If it's a dimer, there's a 4 binding sites. It's IgM pentamer, there's a 10 binding site. So this is a more of a combined strength of multiple affinities, which is non-covalent bonding, but it's like high affinity. That we call, Anho?
Avidity.
Yes, we call avidity. Okay, so you have to understand the differences and importance of this. And there are antibody classes, or we call isotypes, and it has a different role. So first of all, the most abundant antibody against invading pathogen. So it also has a memory function. Anho, can you?
IgG.
Exactly. And then very important for COVID infection. So because our mucosal surface, including all our GI tract and nasal cavities, we have this dimer called IgA. Yes, mucosal area. So this is like secreted from our mucosal cells. But what about the B cell surface? And so when the antibody first come out from the B cell, this actually has this pentameric form, so called a IgM.
IgM.
Exactly. So this has a very high avidity. So that's the first antibody. And the other antibody is much less amount and less well known. But I think so there are questions about allergy. So allergy binds to allergens is actually IgE. And the also a less well understood is a B cell activation rather than IgD. So how does mom provide immune protection for babies? So this is all for like maybe for those who will have a baby or a mom to be mom. It's important for the mom. Mom produce milk, and milk is secretion. So milk has a lot of IgA, and that gives the baby to a protection from the mom. So it's a very important to give the baby initial immune development from mom's milk for as long as possible. I guess it's important, good, beneficial for the baby. But the mom provides immune protection for baby, yes. And also through this placenta blood, I think some of these antibodies are giving into the baby so that the baby also develop their protection. And antibody structure, this is a reference only. I think some information here is useful. Is IgM, GAED. What matters is you can see this total antibody in blood serum. You can see majorities are IgG, and IgA is also 13%, which is remarkable. But IgE is very, very little, and along with IgD as well. So that's something I think useful to know. And regarding to, I just moved around to lymphatics. Firm's question, why do some people face allergic symptoms when they are exposed to something like pollen or eating something like peanut allergy, milk allergy, egg allergy? There are many kinds, but not to others. Okay, what happens to their immune system? I think this is a very fair question, and also I think this allergy has not been resolved. That's why some kids still have to be very careful or can die from this allergic reaction, because if it like swells the neck, and one can die from suffocation, right? So it's very important. So I think Manuel has a good answer for that. So immune system react and attack. Like cytokine storm, if there's a hyperreactivity that originally try to protect us, but then may harm us. That's a big issue, right? So immune system can be hyperactive, producing excessive strong response to foreign entity. So for example, pollen, it's harmless, but this is foreign. And for some people, like really react to block the nose, and many produce excessive mucus and suffer from fever. And these can be hereditary and maybe specific to. So this can be a genetic aspect. And I think, let's see if there's anything like, okay, so I think that's fine. Let's move on to, yeah. So now let's go through a little bit of, so what are the functions of antibodies, typical immune system, the protection mechanisms of our antibody. So one thing is when antibody binds to the invading pathogen, they actually triggers our original defenses and called the complement.
Sorry, could you say that again?
Sorry. So the complement, the name's a little weird, but the complement is actually our innate system. It kind of comes in and tries to make a pore onto bacterium and lyses, just kills it. Okay. And also, as you see, this another thing that causing inflammation and disrupting the cells, and that's relevant with antibody. And also, as I said, so antibody is a kind of a messenger tool towards, let's say there's an invading one, whatever that is, antibody covers it, then that is a marking to our body's immune system. Hey, there's something which is foreign, non-self is there. Try, check it out. So that we call antibody dependence. So our, let's say, immune cells, which is come in and kill the cell. So we call ADCC, or antibody dependent cell cytotoxicity, okay, to target cells. Okay, and the other antibodies, it's because of this multivalency, at least it can, IgG has 2 arms, it can grab this bacterium, or some IgA has 4 arms, it can grab it. So that's called agglutination. So that probably block the functioning of this bacteria. And the other aspect, the antibody can serve as a source for food for big eater cells like macrophage or phagocyte. So that like antibody is putting as a source for this bacterium or virus, such that phagocyte to recognize and come in. So that's why you see this outer parts, so-called FC, constant fragments, is important for the recognition of this phagocyte. All right, so and the other is, of course, it covers the virus or bacterium, so it neutralizes the function of this invading virus and bacteria. So hopefully that make a good sense, and that we call as a neutralization. And another very important aspect, it activates effector cells. Effector cell meaning that the cells actively responds to a stimulus and effect some change, especially immune cells. For example, plasma cell is an effector B cell because it produces antibody. Effector T cell is actively responds to stimulus. Also, master cell is called effector cell. And as I said, a very innate immunity, we activate a complement system. The name is coming from because it complements the ability of antibodies and phagocytes to clear pathogens. So it's an innate system. So, and this is the last slide. This is reference for, but you guys should know at least a key aspect of this. So the 5 classes of immunoglobulin, IgM, this is the first circulating antibody, and the showing IgM indicates a current infection. So I think this is important for disease status detection. IgG is the most abundant in the circulating antibody. IgA is produced by a mucous membrane. IgD, I don't even know much. This is related to B cells, so I'll skip it. But IgE is causing an allergic reaction. And somehow complement system is the ending opsonization. Oh yeah, I didn't say about this. Opsonization is like putting a sauce on that, I think, here. So opsonization, we call this term coding antigen with antibody. It's like putting a sauce, so it makes it more yummy for the phagocyte. And that's, and also, so how the complement system work is a very important discovery. And we call that membrane attack complex or MAC complex. So I think it's useful to watch. Let me take a look. Okay, so. Of animation. Complement has like 9 subunits. You can see. So this is like a pathogen, and they make this one. And so they make different codes of bacteria with the complement. And then what they do is the macrophage actually recognize and phagocyte, and they will become the lysosome to be digested. And next is the membrane of that complement. This pathogen surface is complement from these, then first stage, 3, 2, 5, 6 types of complement. 5, 6, 7, and 8, and 9. So they actually make a kind of a hole here. So it actually ruptures the. So whenever I see these movies, I kind of, it's fascinating that how our body works for this. All right, so that is a today's class. Oh, I think I'm gonna finish at 10, but we have some time. So why don't you, those who haven't asked a question for this, I'll give you 1 minute to write down question, and I'm gonna finish in several minutes. All right, those who have the extra questions, write your question down now.
교수님 마이크가 꺼져 있었어요. 마이크가 꺼져 있으세요.
Oh, right. Okay. Thank you, Jung-Yoon. Yeah, sorry about that. So I was talking about the mRNA vaccine, how important it is nowadays. So understanding different type of vaccine now is equivalent because our humans civilization now, we have a vast vast effort to cope with this COVID-19 vaccine. So there are all different type of vaccines, such as component viral vaccines and whole virus vaccines, and now also mRNA vaccines. So you can actually find many different kind of good material to looking at this. In fact, this could be relevant to our project. And there are questions by IgG level and COVID infections. I think you can also find that out. And also regarding to Jenny's question about why some people are getting this allergy to specific, let's say peanut or whatever it is. And that depends on trigger substance or known as allergen. And the reason why are we different allergic to different things is depending on, as you remember, that's an IgE type E, which makeup is different in every individual, and the people with allergies will typically have more of these antibodies. Okay, so those who have more IgE towards certain allergens, that person is a tendency to develop hyperreaction for that. So this is a hereditary, so confused IgE antibodies are passed on. Okay, that there are something, I think more interesting question regarding to this is in fact, like this is my personal experience that when there are countries which are too clean countries may have more allergies. So we can actually ask this question. So too clean environment causes more allergy to people. So let's get into this. So whether that's true, research has shown children who raise a very clean environment actually more likely to develop fever, asthma, and allergies. Third, hygiene hypothesis. And so, yeah, I think this is not only theory, there's a hypothesis, is a worldview that is testable, right? But let's take a look at different views. Increasing allergies is not from being too clean, just losing touch with old friends. There are like different aspects. Can you weaken system by being too clean? Okay, so this is relevant. Despite the rumor on social media, there's no scientific evidence to suggest that temper stepping a cleaning game is dangerous to immune health. Okay, so this is 2 years ago, so it's quite interesting. So I guess so the people can argue that being dirty is good for your immune system. It says doesn't help your immune system, generally makes inflammation worse, much worse. There's something very wrong with the hygiene hypothesis. Okay, we can see hygiene hypothesis is like controversial. So yeah, I can really say, you know, science is about thinking, producing hypotheses, and see if you test it and see if it's true or not. So you can even see this question of hygiene hypothesis, whether in the end, actually time will figure out whether this is more true or less true. All right, so I think today, as I said, I'm gonna finish it a little early.